Hello everyone, welcome back to the very new video of our channel. So today in this video we'll be completing the chapter 8, Cell the Unit of Life. So without any further delay, let us start the chapter. Okay, so basically here when you look, the introduction starts by asking a question that what is the difference between living and non-living organisms. Okay, so here inanimate means non-living things. So the only difference that lies between them is that living organisms are composed of cells, whereas non-living organisms, they are not composed of cells. So the answer to this question is the presence of the basic unit of life, the cell. Okay, so we know that we have been studying about the cell in all our lower classes, so I'm not going to in detail explain the introduction part about the cell, since we all have studied about this. Okay, so we also know that organisms, if they are composed of single cell, we are called them unicellular organisms, and if they are composed of many cells, they are named as multicellular organisms. We all know this. So here in this paragraph, we briefly discuss about unicellular organisms. See, unicellular organisms, here it is told that they are capable of existing independently and also performing all the essential functions of life. First of all, let me tell you why they are capable of existing independently. Unicellular organisms, single cell, right? Unicellular organisms, consider this to be a single cell, okay? And this is the nucleus. So if this is a single cell, then of course, this cell will be performing all the functions, maybe digestion, excretion, reproduction, respiration, everything will be performed by the single cell. When we talk about multicellular organisms, if I consider the example of a human being, then we have different organs to perform different functions, okay? Not every function is maintained by a single cell. Since we are made up of so many cells, there is a division of labor seen. But in unicellular organisms, can be there a division of labor? Of course not. So this cell performs all the activities by itself. So it exists independently. It does not depend on any other cell. Okay? I hope this point is clear here. That is, unicellular organisms, they are capable of independent existence, since a single cell is found in unicellular organisms. Also, they perform all the essential functions of life. Though unicellular organisms possess a single cell, even then they perform all the essential functions of life. Only then the cell is living, right? Okay, for that reason, this is mentioned over here. Next, let us continue with this. Hope you all are active and are listening to the lecture. Anything less than a complete structure of a cell does not ensure independent living. We all know that a cell consists of cell organelles, cell wall, cell membrane, nucleus, right? So if there is a lacking of any of the following things, then the cell is not complete. Though we know that prokaryotic organisms do not have cell organelles, they do have cell membrane, they do have DNA, they have nucleus, right? So anything less than a complete structure of a cell. So if the structure of the cell is not complete, if plasma membrane, cell membrane is not present, cell wall is not present, nucleus ain't present, then it does not ensure independent living. That is, then the cell cannot survive independently. So hence, this is the point which we'll be studying always. Cell is the fundamental structural and functional unit of all living organisms. Now let us jump into the discoveries. Kids, if you want to speed up the lecture, you can listen to this lecture in 1.5x or 2x, the way you're comfortable in. When we talk about the discoveries, Anton von Leeuwenhoek, he was the first to see and describe a live cell. The word live cell becomes very, very important here because he discovered a live cell. Robert Brown, he later discovered the nucleus. But we have studied, guys, if you remember in your lower classes, you have studied that the discovery of the cell was done by Robert Hooke. Is it false? No, it is true. Robert Hooke, he was the first person to discover the cell, but he discovered a dead cell, okay? The cell was not living. The living cell was discovered by Anton von Leeuwenhoek. The dead cell was discovered by Robert Hooke. The first person to discover the cell was Robert Hooke. For further information, I'll just mention the years in which it was discovered so you people can get a clear-cut idea. Robert Hooke, he discovered the dead cell in plant cord in 1665. Anton von Leeuwenhoek, he discovered the live cell in 1674. And Robert Brown, he later discovered the nucleus in the year 1831, okay? Later, as advancement happened in the field of education, electron microscopes were developed which revealed all the components of the cell, and we are studying the structure of the cell so clearly today. Okay? And now when we talk about the cell theory, two scientists together formulated the cell theory. In 1838, Matthias Claden, okay, he was a German botanist who study about plants. Of course, he will examine plants. He studied a number of plants and he gave the statement that plants are composed of different kinds of cells which form the tissue of the plant. See, when I read this, you be like, what? This is so common, everyone knows this. But at that time, this was a major discovery which had happened. So in 1838, Matthias Claden, he was the first person who gave this statement, okay? Another scientist called Sean, during the same time, 1839, that is after a year, he studied different types of animal cells since he was a British zoologist, and he reported that cells, they had a thin outer layer, okay, which today we recognize it as plasma membrane. So he gave a report that there were thin outer layers present in animal cells. After that, he was also interested to do some discoveries on plants actually. So he studied some of the plant tissues, and he said that the presence of cell wall is a unique characteristic of plant cells. Okay, so both of these was given by Sean. So on this basis, Sean alone, okay, this guy alone, he proposed the hypothesis of cell theory. Hypothesis is a guess, okay. So first scientists give a guess, which other scientists together they take time to prove that. Once it is proved, it is converted into a theory. So first Sean, he proposed the hypothesis that bodies of animals and plants are composed of cells and products of cells. So this hypothesis became a theory later. That's the reason we are studying all this today. So after many experiments conducted on this hypothesis, Cledden and Sean together then they formulated the cell theory. Though the cell theory they formulated together, the hypothesis was only given by Sean. So this is a very important point to be noted also. After the theory was given, this theory did not explain how new cells were formed. It only talked about the composition, like organism is made up of cells and products of cells. Apart from that, it never talked about how new cells were formed. So this problem was noted by Rudolf Virchow in 1855. Then he gave the statement that cells divided and new cells were formed from pre-existing cells only. This is called as omnicellula e cellula in Latin. So this was given by Rudolf Virchow. Okay, so he is given the credit to modify the hypothesis of Cledden and Sean, and he was the person to give the cell theory a final shape. I hope this is clear. So today, cell theory is understood as all living organisms are composed of cells and products of cells. All cells arise from pre-existing cells only. So this first hypothesis was given by Sean, which was later proved by both Sean and Cledden together, the first point, whereas the second point, it was added to the cell theory, which modified and gave the cell theory as a final shape, which was given by Virchow. So these are the points which people should remember and study. Now let's jump into the chapter, the most important points to study here. Okay, now we know that there are two types of cells. Okay, we have prokaryotic and eukaryote cells. In prokaryote cells, it basically lacks a specific cell nucleus. Okay, nucleus wall, that is the nucleus membrane. It lacks the nucleus membrane, whereas eukaryote cells, they have a very well developed nuclear membrane. Okay, let's continue. When we examine an onion peel or a human cheek cell under the microscope, we'll be able to observe the cells. Okay, so this experiment your teacher might have done to you in the lab. So onion peel is considered as a typical plant cell. Can be asked in your MCQs. Okay, the onion cell is a typical plant cell. So since it is a plant cell, of course we find cell wall there as its outer boundary, and within it is its cell membrane. This was about a plant cell. And when we talk about a human cell, its outer membrane is the cell membrane, which is as the delimiting structure. Delimiting is the final structure of the cell. The outermost structure of the cell is cell membrane here, whereas in plant cell it is a cell wall. So inside each cell we have a dense membrane bound structure called nucleus. Nucleus is very dense, and also nucleus is the largest organelle found in a cell. Okay, so inside each cell is a dense membrane bound structure called the nucleus. This nucleus, it contains chromosomes. If you guys don't know about chromosomes, we'll study that in our upcoming lectures. Please don't get tensed about this. I guess most of you know. Okay, nucleus, it contains chromosomes. Chromosomes in turn, they contain the genetic material DNA. What is the full form of DNA? Deoxyribonucleic acid. Okay, also cells which have membrane bound nuclei, we call them eukaryotes, whereas cells that lack a membrane bound nuclei are called prokaryotes. In both prokaryotes and eukaryotic cell, we have a semi-fluid matrix called cytoplasm, which occupies the volume of the cell. Okay, cytoplasm, it occupies the volume of a cell, and it is called a semi-fluid matrix. It is a fluid. Okay, most of the cytoplasm is water. Cytoplasm consists of water, mostly water, and other materials also, but most of it is water, like 72% is water. And when we talk, it is called a semi-fluid matrix. Okay, listen, further I'll clear the doubt about semi-fluid matrix. The thing is, if this is a cell, okay, let me draw this as a cell, and if the interior of the cell is covered by cytoplasm like this, okay, though the inside of the cell is covered by cytoplasm, we might have many organelles. Okay, because of the presence of the organelles, the cytoplasm, it becomes a little less dense, and it is called as a semi-fluid matrix. Okay, it is called as a semi-fluid matrix only because of this reason. So I'm so sorry. So we have a semi-fluid matrix which we call the cytoplasm, which occupies the volume of the cell. Also, the cytoplasm, it is the main arena or the main place for cellular activities to happen in both plants and animal cells. The cytoplasm is the place where chemical reactions occur to keep the cell in living state. The chemical reactions are nothing but the metabolic reactions which happen within a body to keep the cells in a living state. Okay. Besides nucleus, the eukaryotic cells, only the eukaryotic cells have other membrane-bound distinct structures called the organelles. So what are organelles? They are membrane-bound distinct structures. See, the different types of organelles present in eukaryotic cells are endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, microbodies, and maybe some vacuoles. Whereas the prokaryotic cells, they lack such membrane-bound organelles. Why do prokaryotic cells lack organelles while eukaryotic do not lack? The main reason for this is prokaryotic cells are the primitive cells. Primitive in sense, these cells were found in the beginning when the earth was formed. During that time, prokaryotic cells were there. At that time, prokaryotic cells were not that advanced, because of which not all the membrane-bound organelles were present in them. So further, when we study the meaning of prokaryotic cells, this doubt will be clear to everyone. Okay, let us continue. So ribosomes. Ribosomes are also a type of cell organelle, but they are non-membrane bound organelles. That is, membrane is absent here, and it is found in both eukaryotes and prokaryotes. Okay. In prokaryotes, there are no membrane bound organelles. See, when we talk about the nucleus, it is also membraneless, and because of that, there are no other membrane bound organelles. Only ribosome is present in prokaryotes. It is present in prokaryotes because it is membraneless. Okay, ribosomes are not only found in the cytoplasm but also within two organelles, chloroplast and in mitochondria, also on rough endoplasmic reticulum. This will discuss further detailedly. Okay. Animal cell, it also contains another non-membrane bound organelle called centrosome, which helps in cell division. Now, totally, we have studied that the non-membrane bound organelle is ribosome, and another one is centrosome. Centrosome is only present in animal cells, not in plant cells. Okay. Also mentioning that ribosomes, they are found in the cytoplasm of the cell. They are called as protein synthesis machinery, which we'll study further. Also, ribosomes, apart from the cytoplasm, they are also found within two organelles, that is the chloroplast in plants, mitochondria, and on rough endoplasmic reticulum. Also, the cells differ greatly in size, shape, and activities. Example, mycoplasma. Okay, mycoplasma, it belongs to the kingdom. Which kingdom? Monera. Okay, so it is a type of bacteria. They are the smallest cells known, and they are 0.3 micrometer in length. See, the length point is important. So in length they are 0.3 micrometer. Okay. While bacteria, it could be 3 to 5 micrometer in length. Again, we are talking about the length. The largest isolated single cell is the egg of an ostrich. The largest isolated single cell is the egg of the ostrich, and among multicellular organisms, the human red blood cells, RBCs, or the erythrocytes, they have 7.0, that is 7 micrometer. They are 7 micrometer in diameter. So you got the diameter, which is 7 micrometer. So from here you can also find the radius, that which is 7 by 2 micrometer. So you get the radius also. So if there is a question asking regarding the radius of the erythrocytes, then you should be able to answer this question. Also, nerve cells, they are some of the longest cells. Cells also vary greatly in their shape. Cells, they can be disc-like, polygonal, columnar, cuboidal, thread-like, or even irregular in shape. Irregular is something like this, okay? They can also be irregular in their shape. The shape of the cell, this is a very important point which you people should note and also make a note of it. The shape of the cell may vary with the function they perform. I'll show this with the diagram. Here you can see different kinds of cells. So the first thing is the human red blood cells, RBCs. They are round and biconcave. So they are round and biconcave specifically because they have to carry hemoglobin. Okay. So that's a particular thing when we talk about structure and function. See, when we talk about the WBCs, the white blood cells, they are amoeboid in shape because they have to move around the body, and if they were rigid, they couldn't have traveled. That is the reason they are amoeboid or irregular in shape, so that they can pass through the cells easily and attack the foreign particle. When we talk about columnar epithelial cells, they are columnar, they are long since they provide protection and other stuffs like that. They are long and narrow. Nerve cells, they are branched and long. Why aren't nerve cells round and biconcave? They are not round and biconcave because the nerve cells, they actually should pass on the information in the form of impulse. So if nerve cells are long, of course they can what do you call it, they can pass the information very quickly. They are in the form of threads for that. So it is told that the cell structure depends on the function they perform. Okay. So there are other examples also. Trachea, they are elongated, and also the mesophyll cells, they are round and oval. So the mesophyll cells, let me tell you a point about these mesophyll cells. They are parenchymatous cells. Also they have many chloroplasts in it. You see these black dots, they are chloroplasts, which helps in photosynthesis, right? For more understanding, here I have brought a typical prokaryotic cell, which is a bacterial cell. Let us start naming this. So the outermost, okay, the outermost here is the cell wall, and this is the cell membrane. Okay, inner to the cell wall lies the cell membrane. So you can see here there are no other cell organelles, only we have cytoplasm. So this is the cytoplasm, the empty place which occupies most of the volume of the cell. This is the double standard circular DNA which is found in the prokaryotes, and this is the nucleus. Of course, see, this is the nucleus, and as you can see, the nucleus is not enveloped by any nuclear membrane. We have ribosomes. The red dots here represents the ribosomes. Also you can find, you can see some ribosomes which are attached to the cell membrane, right? Some of the ribosomes are attached to the cell membrane also. The blue color structure here is known as the chromatophore. This chromatophore, they are specialized structures which contains pigments. See, this chromatophore is blue in color. Chromato means color. The meaning of the word chroma or chromato means color. Okay, so chromatophore, chromat. So this is the chromatophore. Chromato literally means color, and this is a special extension from the cell membrane. You can see this is an extension from the cell membrane here. So this basically contains pigment, and it helps in photosynthesis. So this is a special extension of the cell membrane. Also, this is also a special extension of the cell membrane. We call it as mesosome. So mesosome, it is a specialized structure which actually helps in many things like cell division, respiration, increases the surface area and all. So the mesosome, we have three types. Okay, this mesosome is present in three times. It can be in the form of a vesicle. Vesicle is something round. Okay, it can be in the form of a vesicle, in the form of lamellae, so this is lamellae, and in the form of tubules, like long as tubes. So this is tubule which I have drawn here. This is in the form of tubules. Okay, just for your understanding, I am making all this. Also we have special, okay, special some extracircular DNA apart from the main DNA. See, this is the main genomic DNA. Wait, let me make it with black. This is the main genomic DNA. Apart from main genomic DNA, we also have small circular double-stranded DNA. We call this plasmids. Okay, plasmids. So this plasmids, we basically, they are used as a vector in genetic engineering, about which you'll study in your class 12. For now, you chill, just know that these are plastids and they are external chromosomal DNA. That's it. How do you store food in this prokaryotic organisms? This is a single cell. This is prokaryotic since it doesn't have a specific nuclear membrane to its nucleus. So how are food stored here? Foods are stored in the form of inclusion bodies. Okay, the food is stored in the form of inclusion bodies. So we have different inclusion bodies. These are membraneless, that is, they are not covered by any membrane. So these blue dots here represent the storage of food in the form of inclusion bodies. Also, you can see the flagella here, which helps in the motility, of course. Also, there are other structures called as pili and fimbriae, about which we'll study later. But pili, they are present on the cell wall in such way. Can you guys see that? These are the pili. And fimbriae, they actually sprout out like this, like the bristles on your toothbrush. Also, we have different shapes of bacteria. They can be round, they can be rod shaped, they can be like comma shaped. Okay, see, this is round, long, rod shaped, comma shaped, or they can be spiral in shape like this, they can be spiral like this. Okay, so all about this, we'll now jump into the ncert. Okay, so the prokaryotic cells are represented by bacteria. Blue-green algae is nothing but cyanobacteria. Okay, blue-green algae is cyanobacteria. Mycoplasma. Mycoplasma is the smallest living cell, 0.3 micrometer in length. We just studied right now. And pleuropneumonia-like organisms. PPLO is nothing but mycoplasma again. Okay, that's the smallest bacterial cells. So they are generally, they are smaller and multiply more rapidly than eukaryotic cells. Known fact. They vary greatly in shape and size. So we have four shape and size basically. They can be bacillus rod shaped, coccus spherical shaped, vibrio comma shaped, and spirillum spiral shaped, the shapes which I showed you earlier. The organization of the prokaryotic cell is fundamentally similar, even though the prokaryotes exhibit a wide range of shapes and activities. This simply means that though there are so many types of prokaryotic cells, you just saw that there is mycoplasma, there is there are cyanobacteria, blue-green algae. Though they perform different functions, some are photosynthetic, some lack cell wall, some are very small. Even then, though they exhibit a large variety of shapes and functions, they have very fundamental arrangement, that is, they have the same double stranded DNA as I showed you earlier. They have ribosomes, everything, the same stuff is there. Okay, so the prokaryotes, all prokaryotes, they have a cell wall surrounding the cell membrane except in mycoplasma. So mycoplasma is an exception. Also, the fluid matrix filling the cell is cytoplasm. This thing we already studied, that the volume of the cell is occupied by cytoplasm, but the difference is there we studied semi-fluid matrix. Okay, in eukaryotes it's semi-fluid matrix because most of the cytoplasm, because compartmentalization is seen, that is, most of the space in the cytoplasm is occupied by cell organelles. But in prokaryotes, since there is no other cell organelle except nucleus and ribosome, most of the cytoplasm is empty, that is, it is fluidy in nature, it doesn't lose its dense nature and its fluidity, that is, most of it is still water. Hope I'm getting clear here. In case of eukaryotes, the cytoplasm is semi-fluid in nature. In case of prokaryotes, it is fluid matrix. Okay, so yes, of course, in prokaryotes there is no well-defined nucleus. Genetic material is naked, not enveloped by nuclear membrane. addition to the genomic DNA. Genomic DNA is the main DNA. The genomic DNA is also known as the single chromosome or circular DNA. So bacteria also have small circular DNA outside the genomic DNA. We call this plasmids. Okay, we studied about this. So this plasmid DNA, it actually gives certain phenotypic characters to such bacteria. Phenotypic is nothing but a character to bacteria. So one such character is resistance to antibiotics, about which you will study in your class 12th. In higher classes you will study that plasmid DNA is used to monitor bacterial transformations with foreign DNA, that is it is used in genetic engineering technologies. Okay. Nuclear membrane is found only in eukaryotes. No organelle like the ones which are present in eukaryotes are found in prokaryotes, except for ribosomes, which we studied earlier. Also prokaryotes, they have something unique in the form of inclusions. Yes, we studied about the inclusion bodies which stores food, okay, reserved materials. A specialized differentiated form of cell membrane. Differentiated, it is a form of cell membrane, not the cell wall. Cell wall is very rigid in nature, only cell membrane is flexible. So the differentiated form of cell membrane called mesosome is a characteristic of prokaryotes. Characteristic of prokaryotes means it's only present in prokaryotes. They are essential infoldings of the cell membrane. So cell membranes foldings, see here, infoldings of cell membrane. Okay, so many people get doubted here. Okay, let me clear your doubt. There, when we studied about the size of the mycoplasma and the size of the bacteria, it was basically the length we studied. Okay, the length of the bacteria was given 3 to 5 micrometer. Here the typical bacteria 1 to 2 micrometer is given. There we studied the length. Okay, see, we studied the length. If this is a bacteria, then the length was 3 to 5. And here they are talking about the size, which is this one. I hope you people get this. So this size up here is given here. Get that? Okay. So here basically we talk about the size. So the typical bacteria is 1 to 2 micrometer in size, that is the top portion, and in length it is 3 to 5 micrometer. PPLO is 0.1 micrometer. Viruses, they are 0.02 to 0.2 micrometer, and typical eukaryotic cell is 10 to 20 micrometer. You people will have to learn this. So about the cell envelope and its modifications, we'll be studying about that in the next lecture video. For more such contents, like, comment and share. If you have any doubt regarding this, please comment down, and also let me know how the lecture video was. Thank you for watching.